It's my great pleasure to introduce our next speaker, Dr. Marissa Bartolomei. Marissa is a professor at the University of Pennsylvania, and I think most of us are familiar with her, also her work, and she has been one of the pioneers in the field of imprinting gene. So she has published hundreds of papers, and she's holding right now multiple NIH grants, including one of the National Center grants, this P50 grants. And then Marissa is going to talk about some of her latest work. So Marissa, are you there?
Yes, I'm there. Sorry.
Okay, great.
I forgot to unmute and show myself here. So let me see.
Okay, all yours.
Okay. All right, and bring that there. Okay, so we should be good. So thank you, Wei, and thank you, Jill, for inviting me to participate. I'm just going to show on the next slide what I want to get through in the next 20 or 25 minutes. And I want to show about how assisted reproduction has been linked to imprinting disorders. But in order to be able to go through that, I just want to define imprinting, tell you a little about in vitro fertilization and the mouse model that we use, and then show how we've linked the phenotypes in the mouse to the procedure. And more recently, I've done a longitudinal studies of outcomes in the mouse, and then give what we are looking for moving forward, because as most of what I'm telling you about is how a very early life intervention can cause something much later. The question is, can this then be transmitted to subsequent generations? So here's the observation that got us very interested in this, and this is some of the documented outcomes of assisted reproduction in humans. And what really interested us was the idea that there were higher than expected frequency of imprinting disorders in humans that were conceived through assisted to in vitro fertilization. So let's just take a step back and say, what is imprinting? Many of you know that, but I'm just to get all of us on the same page. Imprinted genes are expressed from a single parental allele, either the maternal allele or the paternal allele. They reside in clusters, so they're usually anywhere from 2 to dozens of imprinted genes in about a megabase region. And they're regulated by these imprinting control regions, and imprinting control regions are differentially methylated. It's been shown if you delete these imprinting control regions, you have misregulation of all the genes that are linked in that cluster. And if there are errors in DNA methylation, loss of methylation, gain of methylations, you also see misregulation of the genes. One of the really striking aspects of these imprinting control regions, which are differentially methylated in the germline and maintain that differential methylation, is that right after fertilization, when most of the genome is being reprogrammed, these imprinting control regions survive this DNA methylation reprogramming. And this is the period here in which many of the manipulations used in assisted reproduction in IVF occurs. And so the question is, could these procedures affect imprinting control regions? Could they affect other genes in the genome in this early period, causing something much later on in development? So let's just review what the imprinting life cycle looks like. Imprints are established in the germline. So if they're on the maternal allele, they're established in the oocyte. And if they're on the paternal allele, they're established during spermatogenesis. These imprints, as I just mentioned, then escape this genome-wide reprogramming that occurs in that early embryo. And then they must be appropriately erased and reset to go on to the next generation. And this is really is an important area that we're thinking about if certain types of techniques used in assisted reproduction could be affecting these in the future. Okay, and just another reminder again, that there have been a couple imprinting disorders that are associated with in vitro fertilization, including Beckwith-Wiedemann syndrome, which is an overgrowth disorder, and Angelman syndrome, as well as others, including Silver-Russell syndrome. So there are a number of imprinting disorders that are associated with IVF, and in many cases, these imprinting disorders that arise in children conceived by IVF result from loss of DNA methylation of the ICR. Most of the other imprinting disorders normally are associated with defects in the gene, deletions, translocations, but IVF, the vast majority are associated with changes in DNA methylation. So what goes on in vitro fertilization? We know that in vitro fertilization is crucial for infertility treatments, wildlife conservation, but IVF uses exogenous hormones. It uses the actual techniques used in vitro fertilization, extended culture, embryo transfer. There are now more recently freezing embryos, preimplantation, genetic sampling, so removing a blastomere in an early embryo for diagnostic purposes. So there are a lot of technologies that are used in IVF. And these technologies that I mentioned at the beginning occur when these really important processes of genome reprogramming go on. So we have egg production that's stimulated by hormone therapy. The eggs are retrieved, incubated with sperm. They're combined to allow fertilization, cultured for a little bit, and then transferred back to the recipient. And so you have superovulation that occurs to make more eggs, embryo culture, embryo transfer, all of which are occurring at this early reprogramming period as shown here. So superovulation occurs when the oocytes are growing, and that's when DNA methylation is being put on these oocytes. And then embryo culture and embryo transfer, for example, occur during this early period where you have very, very dramatic reprogramming of the genome, DNA methylation erasure, and acquisition of the vast majority of the genome. So the linkage between IVF and imprinting disorders was very compelling to us when we first heard it maybe almost 20 years ago. But the problem is humans are not an experimental model, and it was very difficult to try and understand what was going on. So we and many other groups developed a mouse model of IVF, including John McCary, who's talking later in this session. And what we found in this early mouse model of IVF was that we saw embryonic loss, low birth weight, abnormal placentation, epigenetic aberrations that were actually very similar to some of the things that had been reported in humans. And for an example, here is an E18 mouse placenta in the normal control, and you can see that it's abnormal relative. The two are normal in comparison. So we then wanted to see whether we could figure out which procedures used in art were associated with these abnormal pathology that was reported. So we developed a series of groups: natural matings, embryo transfer alone, where we just allowed the mice to mate normally, removed blastocyst and put back into a recipient, superovulation and embryo transfer, and the whole IVF procedure. Would that involve superovulation, harvesting of eggs and sperm, incubating, culturing to the blastocyst, and transferring back? And so then we took these mice at about term and analyzed them. And I'm just going to briefly summarize what we saw, because this has all been published. And that was, here's the placentas again, natural and embryo transfer, superovulation, embryo transfer, and IVF. And on the left is H&E stain, and on the right is pseudocoloring, just so you can see the labyrinth zone versus the junctional zone. And what we saw was with each procedure, the placenta was a little bit larger, and there was a general overgrowth of the junctional zone relative to the whole placenta. When we looked at DNA methylation of imprinting control regions of a variety of imprinted genes, we saw that IVF, but not these other procedures, embryo transfer, superovulation, was associated with reduced DNA methylation in the placenta. These are 3 imprinted genes, just shown here for an example. So the conclusion of this work that was published about 5 years ago was that with each procedure that one uses in IVF, embryo transfer, you add superovulation, you do the whole procedure, you saw increasing number of abnormalities. So just with embryo transfer, you see a weight change. Add superovulation, you see junctional zone overgrowth. And when you start to do the whole procedure, you see these other abnormalities. So we didn't stop there. Lisa Vrooman in my lab wanted to look much more carefully about what was going on in IVFs to really start to get at the mechanism. And one of the things that she did was she added really a group that tried to isolate embryo culture. So it didn't involve the actual IVF procedure. There was superovulation, but we took the embryos out at the 1-cell stage, cultured to the blastocyst, and then transferred back. And then the second thing she did was also do a time course. And that was because we were mostly at this point looking at term. And so we wanted to look during mouse gestation. And this just shows a timeline where you see the formation of the placenta about mid-gestation. The placenta then grows, and then you see the majority of fetal growth that occurs late in gestation. So Lisa then looked at all of these groups that she defined here at a variety of time points. Now this slide shows is a lot of data, but I'm just going to tell you a couple just very brief points. And the first point is when we looked at 12.5 days of gestation at the fetal weight, we were surprised to see that the fetus was really undergrown at this point, whereas the placenta had a normal size. You didn't really see any difference between the natural and the different groups, so that you could see that when you do the fetal to placental ratio, or you're measuring fetal sufficiency, placental sufficiency, you could see that the fetus was undergrown relative. When you look at 14.5 days, you start to see that the placenta is growing over. Perhaps it's noting that the fetus is too small and it starts to overgrow. And what's rather surprising is if you look later, these embryo culture groups, the fetus continues to overgrow, or excuse me, the placenta continues to overgrow, whereas some of the fetal growth deficiencies are corrected. So we wanted to look a little bit more into what was going on in the mouse placenta. And what we did was we actually, what I should say is we looked at the labyrinth zone, which is responsible for nutrient exchange. It's the vascular component of the fetus, as well as the junctional zone where the endocrine cells, the glycogen is located. And we then did a series of experiments on this, on the placenta. And one of the things we looked at was the vascularity of the placenta in the labyrinth zone, and we used a marker, CD34, which labels fetal endothelial cells. And what we saw was that here's the natural all the way on the left. And as you go with each increasing procedure, you see fewer endothelial cells and really sort of defects in endothelial cells. And this is quantified in the next slide. So at 12.5 days, you see that there's fewer endothelial cells, and although as time goes on, the placenta makes up for this. So it compensates for this initial vascular phenotype. In terms of the junctional zone, we see that the junctional zone starts to overgrow at about 14.5 days. Relative to naturals. You can see it quantified here. We saw that the glycogen cells are actually overrepresented. And then when we looked at DNA methylation, we did a lot of methylation assays. We saw that DNA methylation was abnormal from the onset, which was not surprising to us. Other experiments we had seen suggested that. This is again the placenta. but we only saw DNA hypomethylation of our various imprinted genes in the groups that involved embryo culture. So the conclusions from these first, this next set of experiments, which was recently published, was that embryo Timecourse revealed that the embryo is initially smaller, which may result in vascular defects in the placenta. We're still trying to understand why that happens. The placenta subsequently overgrows. Embryo culture alone is actually no worse than the IVF procedure, and that epigenetic abnormalities are associated with embryo culture. So this brings us back to the question of then what goes on after birth in this mouse model. And I did point out imprinting disorders, which we could explain in part by some of these epigenetic defects that possibly result from culture. We see them to a much lower extent in the fetus in this mouse model than we do in the placenta, but we do see some fetuses that have reduced methylation at the imprinting control regions. But there are also other effects that have been noted in human. There's hypertensive diseases of pregnancy. Our mouse model suggests that we may have some hypertensive markers. There's cardiac or cardiometabolic phenotypes and other kinds of disorders. So we then decided we wanted to go and look longitudinally. And what our paradigm for this outcome assessment was, what we did was we had two groups, the natural and the IVF groups. We let them go almost to term, and then what we did was we delivered the embryos right before they would have naturally delivered so that we could get the placentas. Mice eat the placenta, so we wanted to use the placenta for a biomarker. So we delivered them and we fostered them. We were very careful about numbers of mice, etc. And then what we did with these fosters, we weighed the mice, we marked them so that they could be tracked back to their placentas. And then we did a series of assays longitudinally on these mice for about 40 weeks. We measured triglycerides weekly, or actually monthly. Body weight was weekly. We measured triglycerides, cholesterol, insulin, and we did glucose tolerance tests monthly. And then right before we euthanized the mice, we measured blood pressure and heart rate and body composition by DEXA scanning. And then with the placentas, we weighed and looked at DNA methylation and other parameters. So I'm just going to give you a little bit of a brief summary of what we saw. This paper is out on bioRxiv, so you can look in much more detail. But what we saw was that we had sexually dimorphic phenotypes, with the IVF females having higher cholesterol at most of the time points we analyzed. Actually, the ones that are colored give you an idea of a mouse that's followed longitudinally, just to show there's a consistency in this elevated cholesterol. This is the summary of everything we looked like. The female mice, I didn't actually show you this, the female mice also are heavier relative to the naturals. But the only other thing that we saw was this elevated cholesterol. We did also see reduced DNA methylation in a variety of genes. This could be slightly underpowered. If we had more placentas, we might be able to see a little bit more of this. So then we went and looked at males. Males actually had higher insulin at most of the time points measured relative to the control natural mating animals. They also had higher triglycerides at most of the time points measured, but we didn't see by the assays that we used differences in blood pressure and heart rate, although that just could be that those are pretty hard experiments to do in the mouse, and we need to do something a little bit more careful. We did, however, see that even though the males were not statistically heavier than the naturals, they did have actually a higher percent body fat at 39 weeks relative to the natural controls, and this was statistically significant. Again, all of this is shown here. Higher triglycerides in the males, higher insulin, increased percentage body fat, and there were some changes in DNA methylation in the placenta. So in conclusion, our IVF model, following these mice longitudinally, the offspring exhibit adverse metabolic outcomes that are actually quite consistent with those identified in human. We think that we need to look in more detail in some of these outcomes, but for the time being, it looks like we have identified them. These phenotypes are sexually dimorphic, and I just can't emphasize enough how important it is to analyze IVF outcomes in humans by sex before we make any kinds of conclusions. And in the paper that's online, we also did profiling proteomics, RNA-seq, et cetera, to try and address what the difference is in the pathways that lead to these adverse phenotypes. And we are now continuing this work to look at the effect of IVF on reproduction, looking in the ovaries and testes, these kinds of metabolic phenotypes. It's very possible we're going to see some, and we already have some early indication. And we also are interested in looking at the next generation to see if some of these phenotypes could be transmitted multi-generationally. But again, that work is in progress. So finally, I just want to acknowledge the people that have done this work. This work was started many, many years ago with Melissa Mann and Rocio Rivera. It was a collaboration with Richard Schultz. And more recently, Eric DeWall did some work and before he left the lab. And more recently, Lisa Vrooman, Eric Ron-Calderon, together with Chris Yi and Yamin, have worked on this model. And the outcomes was done by Loren. And again, this is, as was sort of mentioned by Wei, this is all part of a very large P50 grant supported by NICHD, looking at the effect of assisted reproduction in humans and mice. So thank you very much. And if I have more time, I can take questions, or else we can wait to the end.
So thank you very much, Marissa. Excellent talk. So we do have some time for questions. While people are typing their questions, I will take advantage of I have the microphone, so I'll ask you the first question. Marissa, you mentioned that there's a sexual dimorphism in the male and female offspring. So when you analyze placenta, did you, sort of separate sex as well? So meaning that I'm trying to ask that, is there any sexual dimorphism in the placental defects?
So we actually attempted, we only did a few markers. We did some Luma analysis that sort of looks very globally at DNA methylation. And we did see a correlation with defects in DNA methylation in the female placenta with some of these phenotypes. We didn't see a correlation in the male, but again, I think that we do in other studies see some sexually dimorphic phenotypes in the placenta with a higher powered analysis. So we're mostly trying to correlate the outcomes right now with the phenotypes. And I think there's something there, but I think there's more that we need to do especially with DNA methylation. So.
Yeah, so the reason why I'm asking is that people previously have reported that the IVF tend to lead to a skewed sex ratio, and so the question is that is the embryos that are dying, or is the secondary effects to the placental defects?
Yeah, we have these conversations all the time. Every time we think we see like a sexually skewed, litter, we'll have like 6 males or something like that. And the next time we'll have like 6 females. So I think that we're still trying to determine if that's going on in our mouse model. Others have reported it in the mouse, but we're still in the process of trying to see that. We really don't have good data yet looking at that.
Well, thank you, Marissa. So we have questions from the audience. And so here's the one. And are there other epigenetic mechanisms involved in regulating these ICRs in addition to DNA methylation?
So yes, great question. So the reason we looked at DNA methylation is because it was at the time was easier to look at, but people have found differential histone modifications at ICRs, lysine 9 trimethylation and lysine 27 trimethylation. We like the idea that if DNA methylation defect occurs early, potentially during culture when all that reprogramming, usually it's heritable. And so then we get an idea that we're seeing what happened weeks earlier. So that's why we focused on DNA methylation, but definitely there are other mechanisms involved, and it's likely that we would see that when we did more profiling. Yes.
All right. So another question, do you think that IVF outcomes are worse for placenta, and placenta defects are what is driving problem in the embryo fetus? I think this is sort of similar to my question. Which one is the primary? Is the placenta defects the driver, like the cause of the embryo fetal defects, or the other way around, or maybe like the independent?
So I think definitely the effects are worse for the placenta than they are for the fetus, because you will, if for example, if you look at DNA methylation, you'll see most of the placentas have DNA methylation changes for the placenta, in the placentas in the IVF group, but if you look at the fetus, only a small set of them. And the idea is that there may be more redundant mechanisms in place to control epigenetics for the life of an animal versus the relatively short term for the placenta. So I think that it's definitely that the phenotype is worse for the placenta. When we did the Timecourse and we saw that the fetuses were smaller, we were like, oh my God, the phenotype is in the fetus. But then when we looked at the vascular markers and saw there was already defective vascularity in the placenta, we go back to thinking it's probably driven by the placenta. So, but yeah. Definitively, do we know? Not yet.
All right, so we have one more question. So do you think the phenotype, the genotype of the sperm contribute to placental outcomes you observed, or it's just totally linked to eggs? I think this is like what's the relative contribution between sperm and eggs to the defects you're observing?
Yeah, it's interesting because Melissa Mann has done some work that suggests even just looking at superovulation where there's not other manipulations involved, she sees paternal defects later on. So even with superovulation, maybe somehow that causes something else to happen after sort of a normal mating. I think that we see both because we see loss of methylation in sperm and we see loss of methylation in eggs. I'm really in the camp that I think each one of the procedures contributes a little bit. That's why ultimately the goal is to figure out which ones contribute the most so that you can tip the balance back to having everything be balanced and normal. But I think there's something that's in superovulation for eggs. I think that there's a failure to maintain in sperm. There could even be something that goes on with IVF or ICSI for the sperm that maybe impact the offspring. So.
All right. So last question. I think we have more questions, but it's just time is limited. This is one last question. So what type of potential intervention do you recommend if these phenotypes and demethylation alterations were seen in humans? And do you expect women who undergo IVF treatment to have more methyl donors? So basically, what's your recommendation of the any potential treatment intervention to overcome all these defects?
So again, Jaquetta Trausler, you should read her work, it's beautiful. They've shown if you introduce more methyl donors, you can actually downregulate the methylation pathway. So we have to be very careful with this. I think that a lot of people are thinking, yes, one carbon metabolism is involved in some of these phenotypes, but I also think there's a question um, of over-oxidation, since we're not doing these manipulations under the hypoxic conditions that normally goes on in vivos. So there may be things to look at with respect to oxygen stress, with respect to one carbon metabolism. And I think we have a nice system to test. So thank you,
Right.
everybody.
Thank you,
Great question.
Marissa. This is a great talk. So